Hi! Today I will talk about the cell components of prokaryotes. If you're wondering, prokaryotes and eukaryotes are a little different in their cell components. And today I will talk about not really just prokaryotes, but mostly prokaryotes and their cell components. Usually prokaryotes are unicellular, but often there are some exceptions of some of them being multicellular. But because most of them are unicellular, they actually have a lot of components that help them survive on their own. And in order to survive better, they usually form biofilms, group of bacteria or prokaryotes coming together to live together. And they actually share the different types, kind of like functioning like multicellular, but they're absolutely not multicellular. Because it's just unicellular cells coming together to form a group. Okay, so I will talk about how they are able to protect themselves. Let us begin. So here is my awesome diagram that I drew of prokaryotes. I hope it's awesome. So here we'll begin with cell wall components. So if you study microbiology, or if not, it's okay, because microbiology, we usually talk about prokaryotes, but there are two types of prokaryotes, and we call that differential differences between their gram stain. So according to gram stain, there's two types of cell wall, where one is gram positive, which just have peptidoglycan. It's thick layer of peptidoglycan. And if it's gram positive, then you won't see little dots out here, and you'll just see the cell wall. But if it's gram negative, those bacteria have another layer outside of their peptidoglycan. Technically it's part of their cell wall, but it's called outer membrane if gram negative. So what they have is little dots here, actually extra layer surrounding. That's part of their cell wall. So here, this is actually thick part here, and then between this place, so between their outer membrane and cell membrane, like plasma membrane, is where you can find periplasm. So periplasm is also a periplasmic space. Actually, it's in gram-negative bacteria, and this is a region between cell membrane and outer membrane of the cell, and there are various enzymes for hydrolysis, chemoreceptors, and binding proteins. And this is also where transportation, degradation, and motility is carried out. So it's just an extra space. And now we'll talk about cell wall. So now, generally, the other components are similar in almost all of the prokaryotes. We'll begin with cell wall. So it's actually not found in animal cell. If you're surprised, I'll tell you why. Animal cell does not have cell wall, but you can also find cell wall in plant cells. Now, the major difference between plant and eukaryotes and prokaryotic cell wall is prokaryotic cell wall is mostly made of peptidoglycan. What it is made out of peptides, so like amino acid chains and sugar molecules combined together to form layers. Whereas in plants, they're usually made out of cellulose, and cellulose is actually considered carbohydrate because it's made of sugar molecules chained together. So it's actually a polysaccharide, but it's just something that humans can't digest, and even the cows, but they need help from other enzymes usually found in bacteria and so on. So cell wall, that's one of the components here, and their function is its rigid, strong outer layer, which protects cell from bursting due to osmosis, and it also supports the shell by maintaining the shape. So what does that mean? if you see here, inside of the cell, full of ions and proteins and macromolecules here, so it's highly concentrated compared to their outside, unless you're in like a Dead Sea or something. But let's just say that we're not in Dead Sea, and outside has lower ion concentration, which means that water will like to move in to the cell. So water, because osmosis from low concentration to high concentration, water will go in here and causing the cell to become bigger. So cell expands if water comes in, but because cell wall is a strong rigid structure that's held constant, you can only expand as much as you can only expand to the point where cell wall would allow you to. You cannot expand more than what cell wall has allowed you to expand. So it's like, imagine putting balloon in a box and you blow the balloon inside the box, but you cannot blow balloon bigger than the size of the box because box is preventing that to happen. So that's the same idea with cell wall, and so that's how it actually protects the cell from bursting. And also this, even if the cell is in Dead Sea and their water will go out and causing the cell to shrink, but because of the cell wall, cell shape will be maintained. It's just that inside of the cell wall, cell will be like small or bigger depending on what happens with osmosis. The next one is cytoplasmic membrane. So it actually has multiple names. Plasma membrane, cell membrane, cytoplasmic membrane, they're all the same thing. So it's inside here, this inner layer, and it functions like wall in your room. So imagine inside your room is like inside the cell, and your walls are the one that's protecting what's going on outside, protecting you from outside world. Unless there's, and now door is actually acting like transporter porins or pores and so on. So the function of cell membrane is actually to allow only specific components or like molecules that are necessary for your survival to come in and to excrete waste. It's all controlling that process. So this is a barrier, and for example, by having different types of pores and also like transporter molecules, transporter proteins and so on, it will actually also help you survive from harmful substances like antibiotics. For certain types of bacteria, they actually have like, usually gram-negative, have a lot of efflux pump, and that's what is actually conferring them to antibiotic resistance, because efflux pump actually kicks the antibiotics out. And where it is actually, we don't, it's just about 80 to 120. That's like sort of natural number of efflux pump found, and it was not actually made for that purpose, but it certainly helps microbes from survive by also kicking out some antibiotics. And so that's a whole variety of transporting proteins that can be found on cell membrane. So that's very useful. And now we move on to cytoplasm. So here, this is an aqueous mixture containing macromolecules, ions, proteins, ribosomes, and so on. And these actually fill the dead space inside the cell. So this is like ribosome, DNA here, plasmid, and so on. All the proteins are found here. And big difference between prokaryotes and eukaryotes is that prokaryotes don't have organelles. They don't really have organelles, and even DNA is just exposed. So major metabolism, like metabolic reactions, occur inside the cell, just in the cytoplasm. And having a liquid or aqueous material inside the cell is actually allows transporting much easier. And finally, ribosome. these are found within the cytoplasm, and this is where it's the protein synthesis site. So ribosome, cytoplasm, cell membrane, and cell wall is actually the components also found in eukaryotes. And now we'll get more specific into only components that are found in prokaryotes. So I mentioned that outer membrane is in gram-negative though. There's gram-positive and gram-negative. So, and other stuff are also found in gram-positive and gram-negative. Similar idea. So here, now we get to a really important area, nucleoid region. So here is actually, they don't have nucleus like eukaryotes. The DNA is actually just exposed in this region inside the cell. And it's actually randomly or irregular shaped region in cytosol of prokaryotic cell that's housing DNA and a bunch of proteins inside. And it's an open region where various enzymes, RNAs, and proteins are found for cellular metabolism. And this is another reason why having no nucleus membrane is what allows mRNA, so messenger RNA, and protein synthesis to occur at the same time. So you can pretty much make mRNA while you can make mRNA, and when you have a little bit of mRNA made, then ribosomes can start binding to it and make proteins while you're still making that mRNA. So this is only possible because they are not organelles. Like there's no nucleus here, and it's efficient, and that's another reason why prokaryotes grow much faster rate than eukaryotes often. See, I don't really know all of them, so I can't say definitely that prokaryotes always grow faster, so something to keep in mind though. And now we have plasmid here. So this is a piece of DNA that carries non-essential, often or sometimes essential genes. For example, it could be genes for antibiotic resistance. So this is a extra gene. Actually, they are able to replicate on their own. So plasmid, that's the beauty of having plasmid. And this is another reason why antibiotic resistance build up much faster in bacteria compared to eukaryotes, because they are able to transmit that plasmid between neighbors. So it's like, oh hey, you need some help with that antibiotic something, then I'll give you this gene so you can also survive. And this is another reason why scientists are, or like doctors are having difficulty trying to treat bacterial infections, because they know what we're going to use the next one if certain antibiotic doesn't work, because some have already survived that. So plasmid is non-essential. In some they found that it's in some bacteria they found it to be essential genes, but often these are non-essential, but they are absolutely helpful. Now the next one is flagellum. So exception is that it's actually also found in protozoan of eukaryotes. But just for easier learning, let's say, and that's really the only exception. Protozoa have flagellum, but often it's only found in prokaryotes. So here, this is a long microtubule structure that allows cell to move. So it's like kind of like, imagine it to be legs of bacteria. And the funny thing is, imagine pili. So pili are these little ones. Imagine that to be the hands of bacteria or hands of prokaryotes. So pili are proteinaceous multi-subunit structure, and these are used to attach to the surfaces, epithelial cells, or other cells like in order to form biofilms and so on. And it's an important virulence factor for an infection in many microbes, because if you can't bind to the surface, then you cannot certainly replicate, and you won't be able to cause infection. So pili are important virulence factors. And the second last one is outer membrane, as mentioned. Yeah, I already mentioned that. So it contains various porins, efflux pumps, transporters, which confer bacteria with antibiotic resistance. But really, cell membrane do have some types of transporting proteins, but efflux pumps are often found in outer membrane. Okay, I think I mentioned it sort of wrong. And outer membrane also contains lipopolysaccharide, which is a large polymer that have 3 parts. So lipid A, core oligosaccharide, and O antigen. So detailed structure drawing, you can also refer to my other video or my other blog posting about cell peptidoglycan structure. And finally, periplasmic space here in gram-negative only. This is, you know what, this is a region between cell membrane and outer membrane of the cell. And as I mentioned, this is a place where transportation, degradation, motility is carried out. And various enzymes are found here for hydrolysis. Also, some are necessary for chemoreceptors and binding proteins. That's pretty much the cell components of prokaryotes. And if you need to get full notes, it's actually on my blog. So you can find the link down below. And if you just like my drawing and this is good enough for you, well then, good luck with studying. Have fun.